Start recording.
Welcome, everyone, to StabilityHub's celebration of Stability Day 2026. Those that contribute to the success of this complex function of stability often go unnoticed and underappreciated. This event is to applaud stabilitarians and stability stakeholders at all levels while extending our knowledge in multiple areas. We hope you enjoy it while growing professionally as well. We would ask that you turn your camera on if possible, but stay muted unless you're asked to comment further on a question. Please use the Zoom chat feature to submit your questions or comments at any time during this event. If desired, you can address your question to a specific panel member. In the absence of your questions, we've assembled an interesting array of frequently asked stability questions. This session is being recorded and will be available in the articles and video section at stabilityhub.com. The reason why we do Stability Day during the month of February is because of this man. That is Svante Arrhenius. And he was born on the 19th, and we would celebrate on that day, except that Washington's birthday and the American school systems kind of go into competition with us. So we wanted to safely slide it to when you're all back in your office. And aside from that, if we tried to do it during President's Week, we couldn't get the floats up the hill to reach the ski areas so you could see them. But this is much easier. Svante was a Swedish scientist who studied both chemistry and physics, and Arrhenius became the founding father of physical chemistry. He won a Nobel Prize and derived the Arrhenius equation to predict shelf life and introduced the concept of global warming to the world. We're going to catch up with him a little bit later on in the program, but he's the patron saint of stability, so we wanted to have this event during this particular time. I'd like to introduce today's speakers, panelists. Includes representatives from across the stability world, practitioners, as well as suppliers. And they are Geoff Carr, Pharmaceutical Consultant, Retired Director, Analytical Development at Thermo Fisher Patheon. Geoff Cook, Director of Engineering at Parameter Generation and Control, a Binder company. They design and build environmental chambers for industries where conditions have to be precise. Susan Cleary, Director of Product Development at Novatek International. They're makers of process-driven LIMS and other software solutions for pharmaceutical, biotech, and other healthcare industries. Emily Dickinson, Director of Quality Assurance at Linical, an award-winning global CRO that guides partners through every phase of clinical development. Then Monica Edwards, Site and Stability Manager at Precision Stability Storage, a division of Astoriom, which stores materials safely and securely under strict environmental factors. Maria Krisch, Research Fellow, Physical Sciences, FreeThink Technologies, Incorporated, using world-class science to spread better products to market. They pioneered the concept of ISO conversion for rapid shelf life prediction. Chris Latoz, Manager, Stability Services, Hollister Incorporated. Hollister manufactures quality medical products for ostomy, continence, wound, and critical care markets. Adam Margolis, Vice President of Sales at LLab. LLab delivers integrated compliance solutions that make your operations manageable, ensure audit readiness, and accelerate your journey to market. John O'Neill, yours truly, lifelong stabilitarian, PSDG facilitator, and editor at StabilityHub, which is a service to bring stabilitarians, stability support personnel, equipment, and service providers, academicians, and regulators together to find and share knowledge about the stability process. Walter Routh, founder and CEO of Sagana Quality Consulting, specializing in pharmaceutical stability and cost-saving solutions. He is ex-AbbVie Stability Director and also Associate Editor at StabilityHub. So with that, we'd like to start our parade. A little bit virtual, but use your imagination and we'll put it all together. We wanted to honor you with something that really caught your eye. And then first up for floats is Geoff Carr, our consultant and panel member. And Geoff specializes in analytical stability and has written some articles for StabilityHub. You can see that he's a real go-getter there. If you go back to the previous slide, you can see that he was following this marching band, and now he seems to be among them. So good morning, Geoff. Can you tell us what's new in the world of analytical? What's the burning issues you see today?
Yes, good morning, John, and good morning, ladies and gentlemen. Very nice to be talking to everybody again, and albeit only virtually. Yes, my previous job until about a year or so ago, I guess, was as Director of Analytical Development at Thermo Fisher Patheon. I retired about a year or so ago, and I do a little bit of consulting from time to time. But for the most part, I was working with Thermo Fisher Patheon, and what we were doing there was manufacturing pharmaceutical products for our client companies. We are CDMO. We don't manufacture any pharmaceutical products for ourselves. Everything we do is for clients, so we are totally client driven. My particular role running the analytical development group was particularly exciting because you never knew what sort of project was going to come through the door next. All sorts of different types of products will come along and we need to deal with them, and all of the analytical chemistry problems associated with them we had to deal with, and that's what I found most exciting. I think a big challenge which is coming to the industry now, started a couple of years or so ago, and is probably progressing, is dealing with how much development should you put into an analytical method when you've got a product which is still under development and you don't know whether you're ever going to get to the market with it. So historically, we would follow, if you like, a sort of ICH-Q2 type of route, where you do a quick bit of development and get something which gives some retention of the main peak and does some separations of impurities that you know about. And then do what we would call a qualification, I guess, which is a partial validation to feel that you're happy with the results you're getting, and then stomp on and move it and use it for dealing with experimental samples, IND type supporting samples and clinical trial materials. Eventually you get to registration. You need to have your method fully validated by that time. Can't do that anymore because in recent years we have had USP Chapter 1220, ICH Q14, which now all require that you do quite a lot more development on your method and get to understand what the method's doing and understand probably the chemistry of the materials that you're working with, which I think is exactly how we should be doing the job. But this is probably hitting the industry quite hard because a lot more work has got to go into that early development of a method before you can be using it. And I think this is probably the current biggest challenge for the industry. So that would be, I would find that actually a very exciting thing to be doing, but some company managements might differ on that because it's always a big drive to get into clinical trials as quickly as possible, and you don't want to be bothered with the boring old analytical methods development holding up this sort of progress. So that's what I was doing, John.
Thank you, Geoff. I'm watching for any questions we have for Geoff, and you're welcome to ask questions or any presenter as we go along, especially when their float is in view. But I was going to ask if there's some of these method development situations have recently popped up in any of the compendial books, anything new that's in USP or anything along those lines. I think I saw something that there was a lot of activity there, but I might be mistaken.
There's that guy at the front there that looks as though he's got a question there.
Let's see. I'm watching for that. I don't see it just yet. Let me go back one or two. You saw a hand raised someplace?
It's in the slide that you've got there. It's actually me.
Oh, that guy. Sorry, I'm watching the chat. Well, thank you. That was your question, or you're all set then? I think you're all set. We can go to the next float. Coming into view is Emily Dickinson. And Emily, if you would jump out of that cake and tell us a little bit about what's new in stability challenges and what's hot, especially with regard to maybe statistics.
Yeah, absolutely. And I apologize, my camera's not working for some reason. I can't get it to connect. So you'll just have to look at my face on the screen. So as far as some challenges, it's been pretty interesting. So on the clinical side, I know Geoff mentioned making sure that you've got adequate data to go into the clinic, and that's one of the issues that we've seen a lot lately. So on my side, supporting our sponsors to make sure that they are doing the appropriate stability studies, not just to support shipping to the clinical sites, but to support storage at the clinical sites, because once you get into the clinical space, it's not like your stability chamber. So, there may be storage locations that are not necessarily controlled as tightly as your chambers are. Um, I've got one situation where our IP is being stored in the primary investigator's office. So, of course, controlling that temperature is quite challenging. So, when you're working with your product and you're ready to go into that clinical side, really make sure that you've done the studies to support any kind of temperature or storage sort of want situations that you may come into out in the real world. So that includes doing some of your if you have a very narrow controlled temperature, making sure that you're doing those excursion type studies, and being sure that you can back up any type of excursions with statistics also. So not just your normal 2 to 8, but sometimes using I know we've got FreeThink here, so using some accelerated modeling to support any kind of excursions that may happen at your clinical sites once you get there.
Sounds good. Do you think that it might be on the originator of the drug once they send it out to the clinic to supply equipment for them to do measuring, or is it typically supplied by the clinic? I know there's some people that are new to the process and they might not have been maturing as much as long-term companies and may or may not have the equipment to do that kind of monitoring.
So that's really interesting that you bring that up. So a lot of the times, once you get into the clinic, that burden of ensuring proper storage will fall on the clinical site. So making sure that they have the appropriate equipment to either hold that product at the prescribed temperature, or to monitor the temperature and the conditions where it's being stored. So that's one of the dynamics once you get into that clinical space, is working with your sponsor to make sure that, one, the conditions are fully understood, the expectations of how that monitoring and that storage is going to occur. But if you're working with a CRO like the company that I work for, working with your project team who's going to be overseeing the site too, to make sure that they understand, I hate to call them pitfalls, but the stability pitfalls that may come along with that product, and what would need to be done to ensure that you're not just tossing money out the window if you can't support temperature excursions or different storage conditions.
Would you say any of that data that comes back, since it's not exactly right at 30 degrees or 40 degrees or anything else that you set, is variable? Does that lend itself to, or I should say, doesn't lend itself to statistical analysis? Does that throw it off?
Are you referring to within the patient population, or just related to the drug product itself?
Just related to the drug product itself.
Yeah, anytime you're getting data back that's outside of what your normal conditions are, that's definitely data that you can use to support excursions, whether it's um, to support, shipping from your distributor to your clinical site, any kind of the administration excursions that may um, occur between the point of dispensing and when it's actually being administered to the patient. I will say, in my past life, many lives ago, I worked on a refrigerated product, and it was actually our burden of proof to track the different conditions from the time that the drug product was manufactured until it got into the patient, and make sure that we had adequate data to support any kind of excursions outside of refrigeration. So that's anytime that you get those data points, that can definitely be used to help support the expanded storage condition of your product.
Okay, well, thank you.
Yeah, absolutely.
We promised that we would have bizarre stability stories as part of our program, so we have at least 3 this morning. The first one has to do, ironically, since we just been discussing temperature and humidity in weird situations where it may not be tightly controlled. Wanted to bring up the time in the 1980s, the only chamber fire that I've ever heard of that was started by the chamber itself, as opposed to maybe your plant being on fire. This was submitted by Peter Carlton way back then. He had a stability chamber set at 50 degrees centigrade, and it had a runaway temperature climb. It got higher and higher, and I believe it was not necessarily the temperature that caused the fire, but the temperature that had the impact on the equipment. And some of his controls burst into flames, and he lost everything that he had in the chamber back then. So it points out that it's if you have the technology today to put in a runaway detector and stop the equipment, shut it all down once it reaches a certain dangerous point. And we might ask our representative from Parameter, Geoff Cook, if he's aware of those kind of controls, or are there other measures in effect these days?
Yes. Yes, sorry, having trouble finding the mute button on my Zoom app. So it really goes to the extent you need. We've had customers put secondary and tertiary fire suppression systems in our chambers, just depending on the level of protection you need. A lot of times we'll see the sprinkler heads themselves condense water if the facility's water is cold in a hot and humid chamber. In those instances, we take special steps to make sure that the sprinkler heads themselves are dry fit sprinkler heads. Beyond that, we have had sort of explosive gas suppression systems inside the chambers that will, without water to cover all the product, it will use up all the oxygen in the chamber and put the fire out that way to extinguish the fire without actually getting all of the product in the chamber wet. So there was quite a bit of expense that goes into that. So the level to which you need is definitely a factor.
Well, thank you for that information, and that kind of takes the fear out of our chambers catching fire that would be extinguished quickly if it did. I know a number of us are under regulations that require sprinkler heads in our chambers, and we'll come back to that topic a little bit later on. Wanted to take the opportunity to ask a few other questions here or there. We have some from the audience. Welcome to ask those. If not, I will go over to a question, I think, for Walter. And the scenario was that we frequently have R&D people asking for some of our old purgeable samples. I know this happened to me many times. These are often expired materials that have been in the market. We keep these for several months after a study is completed. What is the risk of sending samples off to R&D for further analysis in these cases? So debated far and wide. On one hand, it's great because you can't find these other samples. On the other, it's got a risk. So Walter, you want to comment?
Yeah, there is the risk of, if they find some really wacky result that was unexpected, that shows that lot may have been behaving differently. However, I think per the question you're saying, it would be sometime after expiry if it was on the market. So ultimately, the risk is very low. I mean, you may find a weird result that causes you need to look into and figure out why that happened. But I don't even know if you would need a deviation because, again, you're talking about something beyond expiry. You don't know if that occurred past expiry, when it occurred, could have occurred in the R&D area. So with all that, there's enough extenuating circumstances that I believe the risk is reduced. It was, for us, it was reduced enough that we did do that frequently. We did build in our procedures specifically, we called those non-routine sample requests. We allowed it, and we gave some conditions under which they couldn't do it, namely if it was still in the market and not expired, or if a very similar batch was still in the market or something like that. So we had some rules around it, and ultimately, though, still the risk is quite low. The risk of detection is even lower, I think.
Yep. I think it's a good point about the rules. You need to make sure that it can never be circulated back into the laboratory for alternative results to some other poor ones that happened. I think that's where the FDA comes in and wants us to have some really good explanations, controls, and I think you described that nicely.
Mm-hmm.
Let's ask another one. I'm watching to see if anything's popping up in the chat. We had a strange month in which several prolonged existing of several hours worth of excursions occurred in our 4075 chamber. A preventative maintenance, a cleaning, a brief outage, and a reorganization and inventory. Overall, the chamber was below 35 degrees C for nearly 24 hours. We don't track cumulative excursions, but it seems like this month could catch an inspector's eye. If they received our monitoring data, should I consider initiating a deviation for this strange month? A really good question. And lots of times we'll see people asking, what about cumulative excursions? When we get to the FreeThink part, we'll ask Maria to comment on that. But I think it's not necessarily a deviation unless one or the other of any of these put it out. And a lot of these we can excuse ahead of time and say it's a planned thing. Some people don't like the idea of planned deviations, but we do have planned maintenance and those kind of things that are happening. But I think a very good documentation of all that. And then applying, as Emily mentioned, if you have some data to show that if you spend some time at a different temperature, that you're not endangering your shelf life. In this particular case, it went a little bit lower. And maybe a stability review committee would take a look at that along with the statisticians and possibly add a little bit of time onto that study. Each company will do it a little bit differently. So it's important to discuss with your quality folks and to make sure that everybody's on board with your plan. Let's see, I'm going to hop down a couple of floats and then come back because I don't see our LLab person yet. I know he has a meeting that he's coming out of. So we'll come back. And we are at FreeThink's float. FreeThink Technologies does a lot of great work in abbreviated stability studies and has developed all kinds of technologies around that. So Maria, can you tell us a little bit about what's going on with FreeThink and the projects that they're under development?
Yeah, hello everyone, and thank you for the invitation. So FreeThink is best known for ASAP, which is a type of accelerated predictive stability. The idea is that we do short-term experimental studies, so these are empirical, and develop models. So they're models where it's temperature and RH are the main variables. We're looking at kinetics of a particular drug product. It is empirical, so it's the one that's tested, and try to use that to predict stability in packaging because we have RH as a functional variable. So if you change packaging, you'll change the RH. Now, one of the, I think, exciting things in this area lately has been that the ICHQ1 is under revision. The draft came out, I guess, earlier last year. I think they got a lot of comments and are incorporating those. But for the first time, it's really more directly addressing modeling. And that's exciting for folks like us who do a lot of modeling because I think it's opened some companies up to considering more extensive use of models to support things like, as Emily Dickinson was saying, in-use stability, things like multiple excursions. There's all sorts of things you can do if you have a functional model and you can try different packaging, different excursions, things like that. So I think we've been noticing more projects that are kind of later stage because of this sort of developing Q1 coming out that's addressing the concept of modeling and things like that.
Thank you. How about answering a question for us? What type of products can ASAP be used for? I know it's adaptable to many, but not everything.
Yep. Yeah, so a lot. The most common thing it's used for, I should say there's products and there's things, is chemical stability. There also is a fairly extensive use for dissolution changes, which also model. I'd say the biggest area is, small molecule drug products, APIs, but larger systems also do follow what we call moisture modified Arrhenius kinetics in a lot of cases. So the key things are usually that you need a specific chemical change that is, affecting stability. So if you're looking at a really large system, if it's some sort of simple chemical change that's limiting shelf life, it may really look a lot like a small molecule, as opposed to some big conformational change, which might be more complicated. And the other thing is that there needs to be a quantitative method to look at the change that you're looking at. So especially as you get to larger systems or biological systems, sometimes the analytical methods have much wider error bars. I'd say to get a good model, we generally need something where the error in the measurement is not more than 20% of the difference between your starting level and the specification. If it's bigger than that, it's not that you can't model it, but the model will be so broad that it won't really restrict you from anything. It'll say everything is possible. But to get a good, useful model, I'd say that a quantitative method with reasonable error bars. But given that, lots and lots of different products. So we look at tablets, capsules, gels, patches, soft gels, APIs, all sorts of things.
And folks can license the ASAP program to run in their own shop without necessarily having to send samples in?
Oh, absolutely. Yeah. So we have kind of part of our business is a software business. So a lot of companies license our software, do the projects internally. For companies that don't want to do that, we also work as a CRO and do it. And I'm noticing there's a question in the chat. Yes, there are some good examples on biologics in addition to small molecules. The FreeThink website, we try to collect papers that are published that are relevant. So if you're interested, there's some reference publications. I think we've done one on a larger system, but there's a number of our other users, other companies that have published things. So if you ever want to look at our website, we have some papers listed with larger biologic examples.
Thank you.
Thanks for the question.
Going back to our previous question about running some things on the effect of cumulative excursions. I know I was speaking with some of your staff earlier in the decade, and they said it was possible, but there are certain requirements that client would have to fulfill for the using the accelerated predictive stability process. So can you tell us a little bit about how you would go about gathering the right information to calculate the impact of multiple excursions?
Yeah, so it's actually pretty easy these days. So basically you need an underlying model with temperature and humidity, which is our normal kind of study. And then you basically just have to have an understanding of what the excursion was and when it occurred. That's what you would put in. And you can put in as many as you want. And basically the program's calculating the response of the shelf life limiting attribute as a function of temperature and humidity. So as those change, you'll just be changing those external conditions and then going back. So it really just, I wouldn't say it just adds up because it does matter when they happen because you'll be at a different point. But you can enter them pretty simply as long as you have all that data about when the excursions happened and what they looked like. It's really all you need.
Great, thank you. That's very encouraging.
Yeah.
And then finally, your company, along with LASA Science, is doing the Science of Stability Conference. This year it's overseas. Can you tell us a little bit about that program?
Yeah, so FreeThink and LASA, which is a nonprofit in the UK, co-sponsor a Science of Stability conference, basically just a conference to try and talk about interesting science questions in stability. We have one every year. It alternates between the US and Europe. And this year it is in Glasgow, Scotland. And it is in April 27th to 29th. So 3 days. We're still registering people. If you're interested and you type in Science of Stability Conference in Google, you'll find a website that has, a list of the speakers and the logistics. But it's an interesting conference and, good conversations. So
Very good. Thank you so much.
And I should say LASA is a company that does predictive software that does things like predicting force degradation pathways, what you would expect. So they're
We're up to the Hollister group. They don't have a float, but they have a gnarly looking of a motorcyclist here. Hollister deals in devices, and these are mechanical devices rather than medical devices. Chris Latoz, you represent Hollister as a long-term stabilitarian. Can you introduce your company and the latest challenges that you're seeing?
Yeah, so as John mentioned, we are a medical device manufacturer. So these are strictly medical devices. We don't do drug device combination products. Our medical devices are single use, disposable. So a lot of different polymers, combination of polymers are used. We make things such as intermittent urinary catheters. So spinal cord injured patients, they may have to self-catheterize in order to urinate maybe 4 or 5 times a day. So we make the catheters for that. Then also we make ostomy pouches and accessories. So people that have colorectal cancer that may have to have part of their intestine removed, we make the collection bags to collect waste. So that's what we do. So they're class 1 and class 2 medical devices. Some are sterile, especially the catheters. Most of the ostomy appliances are not sterile.
We had a question. Are there field alert requirements for device testing on stability like they would have in the classic pharmaceuticals? And if so, what are the timing requirements and the reporting structure? Are they different?
Yeah, so the device world is quite a bit different than pharma. So for instance, for stability, we're not required to put an annual lot of, any product on, do an annual stability lot. That's not a requirement for medical devices. When it comes to field alerts, it really depends. So if you had a stability, out of spec result, really it gets down to could it cause harm, to the patient or not? With our devices, especially with ostomy, if we had an out of spec stability result, wouldn't cause harm necessarily. It would not be a good situation to the end user. But unless the out of spec stability result is going to cause is likely to cause or contribute to harm to the end user, there's not a reporting requirement to the FDA. In instances where it would cause harm, the reporting requirement is generally like 30 days once you're aware of the situation. And I think there's special circumstances where you have to report within 5 days. So yeah, we're quite a bit different than drug.
Okay, well, thank you. Any challenges you faced that particularly come to mind for your stability program history?
Yeah, so again, medical devices are quite a bit different than drug. When you talk about stability, drug, you have, certain tests, assay impurity, identity, so forth, pretty well established stability indicating test. Medical devices, it covers a whole gamut, anything from a tongue depressor to a implantable pacemaker. Those, that's the kind of gamut of medical devices. So there's no, it's really up to the manufacturer to determine what the stability indicating test for that particular device are. So that's one of the challenges we face. What are the things that we need to test in our stability program? Related to that, of course, is the acceptance criterion. If it's we do make some skin creams and lotions, and, pH is important. You want to make sure it's not going to cause harm to the, patient's skin, especially as those products age. So we do have pretty established requirements for pH. Other attributes, again, talking about ostomy pouches, these are, pouches constructed of polymer films that are heat sealed together. So we'll look at some of the tensile properties. We have requirements, but is it clinically relevant? A lot of times it isn't necessarily. Our requirements for tensile strength would be way higher than what would be required like in a clinical setting. So that's kind of the challenges we run into for medical device stability.
Okay, well, thank you. We want to go on to our next stability, believe it or not. And that's a story about what happens when the FDA is in-house and they find something, should you try to fix it right away? And my example is that the FDA came to a company I was working at and found 2 or 3 discrepancies in the inventory count just by asking what's in the bin and how many samples are in there. And we were horrified to find them come up 2 or 3 times in a row with a count that was different than what we had in our records. So they called our entire staff together after the FDA left for the evening. And we spent most of the night in all the stability chambers on our hands and knees, spreading out samples, counting them, putting new records into place with the hopes that we could tell them in the morning that we had 100% accuracy in our stability area for inventory. And what we discovered was by taking people and depriving them of sleep and going home when they should have been going home for that day, that was to, let me get on the right slide here. That was to overcome our problem, but it did not overcome it. We ended up introducing 54 new errors into the process. So it's not always the best idea to try and fix things in real time. It's better to acknowledge them, put together a program, announce what the program is, and then follow through. And we learned our lesson on that one. So we don't advise people to try to fix things immediately unless it's really, really simple. But if you're going to try and pull everybody in overnight to work on something, not necessarily a great idea. Let's move on to Novatek International, our neighbors to the north. And Novatek is working here in the middle of the night on their float to overcome your computing solutions. So Susan Cleary, could you tell us a little bit about the challenges you're facing these days in the world of software?
So, well, software I find is like regulations. It's always evolving, it's always changing. And of course, with the new ICH, changes that are coming up, we've been looking at how to evolve our software to keep up with that. And we're finding more and more we have to make enhancements to the system to add in more trending tools and more, shelf life determination tools. Of course, Novatek Systems has all the statistical analysis for stability built directly into the system. So there's never a need to export the data. So that said, we have already the Arrhenius equation, the p-value, multiple regression, ANOVA tables. So all this is built in. So now we're looking at other ways to change it, to add in like out of trend tools. One of the recent observations we saw was that the company was not comparing the data from batch to batch to batch. So now we've added more enhancements into the system. So our latest version is including out of trend tools to see how you're out of trend. And the trend is determined not just based on that singular batch, but all previous batches of that product. And, also doing these batch comparisons to see if your, degradation is in line with previous batches. So I think that's one of the biggest challenges facing the industry. So enhanced trend analysis, I think, is something that we're working really hard on right now to make sure our clients have what they need to do their statistical analysis on the data.
Okay, and a couple of questions. What's one of the cornerstones for digitalization of stability study management? So we talked a lot in the past about digitalization of major systems and companies. What's specific to stability that would be a cornerstone?
I think the biggest value you can get, a cornerstone really for digitalization, if you're pushing towards that, would be to integrate your equipment with your digitalized solution. So you don't want to do manual transcription checks or manual data entry. We're all human, we all make mistakes. So I find the companies that have the most success with digitalization are the ones that make priority of integrating all their equipment, whether it's your pH meters or your HPLCs or even your balances. All of this information should automatically be fed into your digitalized solution. And of course, for me, this is a cornerstone. Digitalization, it's intended for you to completely skip the paper. So if you have to go take a reading off a balance or get some printout and then go and put it into the system, then you're really defeating the purpose of doing a digitalization changeover. So I think that for me is the biggest cornerstone, the biggest bang for your buck you can get if you're moving towards that type of platform.
Mm-hmm. Hey, when it comes to statistics, are there any particular roadblocks to implementing digitalization of stability statistics?
Yes, the biggest roadblock I see coming up for clients, well, not our clients, is that the statistics are built into the system with the data. So when I say not our clients, they are our clients now. We have clients that are using a different system as their system of record, whether it's your labware or what have you. And we've designed interfaces now that you can actually bring all of your statistics, all of your data into the Novatek system, and then be able to use the statistical packages that are dedicated for stability. So I think the roadblock that a lot of companies out there are facing is they have all of this data in a digitalized solution, but there's no way to analyze it. They have to take it out and put it in an Excel sheet or SAS or Minitab or JMP, one of these external statistical analysis packages. And of course, as soon as you remove the data from where you've housed it from its validated environment, you're diminishing data integrity, you're increasing the risk of mistakes and errors in the data. Compliancy also comes into question. So I think when you're looking at doing a digitalization solution, make sure that your statistical package is built in with it so you don't have to move your data around.
Hey, thanks.
Thank you.
One last one. Is there a practical way to transfer test results or data from a retiring limbs to a newly installed limbs? I know over the course of my career, we tried to do that several times and almost every single time gave up. It was too much of a process. But with the latest in techniques and digitalization, is there a better process in place now? Or are we still hindered from taking old data and sticking it into new systems?
So you have to look at it two ways, I guess. So the first way would be you're retiring a system and you're moving to a completely different software system, okay, completely different solution. Or you're retiring a system because you're upgrading to the same system, just a new version, and you want to move your data over. Okay, so if you're moving it from two completely different systems, of course, you're dealing with completely different data structures. The new company may not be familiar with the old data structure coming from the retired system. So yes, there are a lot of challenges with that. But now more and more, you're right, technology is advanced. It's becoming a lot easier now to extract data from databases and put it into like an XML format or a CSV format, some format where the new system can easily pick it up and migrate it into the new database, the new system. One of the things that you do have to consider, of course, is how much data do you want to bring over? Some stability studies can go 3-4 years. So when we've done some of these migrations, consideration was given to say, okay, we're going to bring over only the studies that are less than halfway finished, for example. And they were going to finish them off in the old system. So you also want to consider what kind of data you want to bring over or how much data you want to bring over. Some companies do want to have all their data brought over because, again, going to that statistical analysis, you have all that old data, you can use it to determine, if your new data is out of trend, and do those batch comparisons that are now becoming more and more prevalent in the industry. From an old version to a new version of the same company, of course, it's much easier. The company has the database structure. They know exactly where the data is and so on. And of course, it's easy to move it over. In some cases, we don't even have to move it into a different database. You just upgrade the database itself. So all of the data is there. It never moves. We just change the structure of the database to match the new version of the system that you're moving towards. Finally, no matter which way you do it, you always have to really give a lot of consideration into the validation of the changes to the database, if it's, remaining in place and you're just doing an upgrade, or of the data being migrated. You have to look at it and do, record count checks, verify you got all the records. So that's quantitative, but you also have to look at it from a qualitative perspective to make sure that you go in and check, your records. Now, if you have, 500 or 1,000 studies that you've done over the course of X number of years, that's a lot of data to verify. So you have to incorporate a risk assessment to say, okay, well, we're going to evaluate, let's say, the square root of n plus 1 number of records from a qualitative standpoint, go in and verify that the old and the new systems both match. So there are some challenges towards doing that. And of course, the validation exercises sometimes become quite difficult. Okay, but it is something that can be achieved. And you're right, more and more nowadays, it's becoming easier and easier to bring it over because it's getting much easier to talk back and forth across digitalized solutions.
We have a chat question that's directly related to that. Maybe that you as a supplier can comment, but I think they're also looking for people with experience from the user point of view as well. How everyone is handling this with different CDMOs. They have different SLIM system for stability data, usually receiving C of A's from them and have to enter the result again in a different system at our end for stability. Anyway, two different companies collaborate in the result transfer. And I do recall those kind of days when was it permissible for them to transfer data directly into your system? Could you go and retrieve it, or could you just go and look at it? A lot of different aspects to that. So we're open to comments on that from both the audience and from Susan.
Okay. Well, speaking as a vendor, we have a software system. Many CDMOs are using it, and we've been doing this for 25 years now. So we've seen quite a bit of, different records trying to be brought into the system. So solutions like allowing an external vendor, an external company to log in and enter the results themselves. That was something that came up numerous times, but now has gone to the wayside because of security concerns. They don't want external people logging into their database and so on. We've done features where you can export a standard sheet, and this is sent out, and then that company will fill it in with all the results and send it back to you, and then it directly imports right back into the system. So that way the company knows what they're testing, which results they need, whether if it's impurity, they have to give the RRT as well, and so on. So it's a formal sheet that they're given to fill in, or Excel sheet, I should say. It's not paper. And then they send that back. We've also had some companies that they get their contract labs to agree on a format of a document. And the document is always that format, whether it be an Excel sheet or an XML or a PDF. And then that is just absorbed into the system. You could upload it, parse it, and then the results come in. So there are a variety of solutions, to not have to manually transcribe data. Digitalization, of course, knows, we want to move away from that. So absolutely, there's ways to handle that. And I don't know, does someone else have something to add to that? Okay.
All righty, we'll move on then. You can either respond in chat or you're able to raise your hand. Any one of those things will do. Let's take a look at this next float. Parameter Generation and Control, a Binder company, and Binder is from Germany. So we've honored them with a Bavarian castle on their float. Geoff Cook, you are from Parameter, and can you tell us a little bit about the latest challenges that you're working on with your company? You might be speaking, but we're not hearing. Oh, I see he was out and I'm letting him back in.
And it looks like he's frozen on the screen there.
Could be. We can always come back to his float. So Geoff, if you can hear us, hop on, or if not, we'll come cycle back to you. Let's cycle back. We'll
Okay.
go to our next.
Okay, I made it.
You're in? Okay, we'll back up to your float.
Sorry about that.
So
As
the
soon as I tried
question,
to unmute myself, I closed out of the app.
no problem. Our question was, what kind of challenges is the world of the makers of stability chambers facing these days and the latest developments?
Yeah, of course, John. So challenges are the ever-changing regulations on refrigerants. The reach-ins have much smaller refrigeration systems, therefore much smaller refrigeration charge sizes, and fall into a much easier category to comply with. So the refrigeration regulations and compliance are always a factor and always changing. But as far as new developments, we are very excited about our redundant systems, having two environmental control units on one room for peace of mind that you'll never have to worry about an out of spec situation. The units alternate on a time base, and also if anything happens to one of the units, the secondary unit will take over and prevent any out of tolerance conditions. We're currently working on optimizing that switchover between the units to actually have the chamber stay within temperature and humidity tolerance during that switchover, which is a delicate dance between two machines. Another thing that we're looking at is continuous mapping of the chambers. Wireless loggers are becoming much more reliable and affordable, and all the AI and software developments are just making it a lot more interesting and attainable. So continuous mapping is something that we are looking at, and the ability to just recalibrate a sensor at the push of a button through a web browser. Yeah, and we're also working heavily on the layout of the ducting in relation to the shelving inside the rooms. You know, the more product you put on shelving, the more you should consider how it's going to affect the airflow, obstruct the sensor, and cause variations in the uniformity inside the room.
Okay, and we have a couple of questions along the lines of the chambers. And one's as simple as someone has a chamber that has a concrete floor that's not insulated. It's a major design hurdle and expense for them. If it's necessary to have the floor set up that way, or the air handle, they can't have an air handler overcome the ambient ground temperature. What's the best solution?
Yeah, certainly. So the cement slabs, it's often that the temperature is the first thing that's considered, but I find that cement is very porous, and we have more issues with the vapor barrier than the actual temperature. So if you have a 4075 room and a cold cement slab, not only do you risk losing some of the uniformity in the room and not being able to maybe use the lower shelves, but at certain dew point deltas, you could end up with condensation on that cold floor. The condensation on the floor can be a safety hazard. It can cause the walls to deteriorate. It can cause the gasket on the bottom of the door to deteriorate and further lend to uniformity issues. So I guess it's kind of is the uniformity worth the cost of the floor?
Mm-hmm.
Interesting.
Okay, thank you. And then people can work with their supplier to overcome those kind of things or get around them if necessary. How about the grade of water used in a chamber's humidification system? How does that impact the equipment? Because a number of people have said that if they use city water, they get mineralization. And if they use ultra purified water, it starts to eat out the stainless steel. So how does someone address the water issue for their chamber's humidification?
Yeah, that's a really good question, John. A question I get almost weekly because it's very dependent on the method of control that you're using. The humidity control means that you're going to need water somewhere, whether you're putting water into the air or removing water from the air. You need processed water or you need a drain. Both of those are important, but this question seems related to the processed water. The processed water is going to have to meet the manufacturer's specifications. Their parameter uses a dew point method of humidity control. We do not have any boilers or steam injection or desiccant. We simply use the laws of physics. And since we do not have a boiler, there is no high purity water needed. Like you said, John, the high purity water, if it's heavily deionized, can actually leach ions from the stainless steel and the evaporator coil and cause the actual metal in the machine to break down. So it really depends on the manufacturer's specifications. For the parameter dew point method of control, standard tap water is often most desirable. RO water is also a good option. It requires that filtration infrastructure. If you have a manufacturer that is using steam injection and desiccant, they have a boiler likely. A boiler means you're going to be boiling water, and using tap water or municipal water is going to leave behind those hard water deposits. So that method of environmental control will often require RO and also the DI water, that filtration infrastructure to feed the boiler without hard water deposits growing inside the room, inside the steam chamber.
Thanks. I got one more. Are there recognizable signs that a stability chamber is reaching its retirement point? When should we take up our pen and go down to the purchasing department and say, hey, I need a new stability chamber?
Certainly, John. I mean, the air handler is similar to a car. As long as you upkeep maintenance on it periodically, we recommend quarterly or twice a year at least. Those signs should be fairly visible. Obviously, components will break down. The manufacturer should be able to provide expected life of the components. I expect a pump to last 5 years and a blower 10, and that's all itemized out in a table based on the part and experience of it. Generally, if you keep up with maintenance, a parameter unit should run 20 plus years, sometimes 40 plus. But the signs of deterioration in the walk-in chamber, you can actually see physical deterioration. Obviously, 4075 is an extreme condition. Even with stainless steel wall panels, after 10 years, you might see some rust build up on those panels. We also see the biggest hurdle for the lifespan of equipment nowadays becoming much more related to the electronics. With the rate of development these days and technology changes, the electronics go obsolete almost after as little as 5 years. It's hard to upgrade the old paper chart recorders. They don't even really make the paper for them anymore. Old circuit boards and the new motors are often smaller. Sometimes they're more powerful, and so the physical size of the new components fitting in the chassis of the older machine becomes an obstacle. But this day and age, it's more often the electronics going obsolete.
Okay, signs we can watch for. I know I asked for stability chambers year after year, and I was never in the right cycle for the budget. Then finally, they could not be revalidated, and everybody said, how could you let this happen? And all I had to do was point back to the purchase requisitions for the multiple years, and that answered their question. Okay, thank you. Let's do another stabilities, believe it or not. And this was a situation where there was FDA audit going on, and the auditors were probably at the top end of the scale for being suspicious of the company they were auditing. They challenged everything. They believed that we were committing fraud at every turn. And this was a very reputable company. And I did not see any fraud. I was in the war room when the inspection was going on. And they challenged us in several different ways. Number one is that supervisors were present in the room when they were questioning employees. And they thought the employees were throwing their answers because the supervisor would punish them if they didn't answer correctly. And I took umbrage with that. But at any rate, we were thrown out of that. And then after a while, the chief inspector came through into the war room where we're processing all the paperwork and checking things before they went out for the inspector to see. And she demanded that we no longer bring anything into the war room, that it come directly from its source to her. And we all looked at each other and said, this is not the normal process that we normally take this through. But we had to shut down the inspection part of the war room. It was kind of interesting, and it was a lesson to us in flexibility, that we started checking documents way down the hallway where they were pulled from instead of going through the war room. They did go direct, but the war room personnel were now distributed to the point of origin. So you have to be flexible whenever these things occur. That was the lesson we learned in that particular experience. Let's take another question. Checking again on our chats. This is for Emily. Should the stability group perform data analysis on every active formal stability study, even when a newer formulation has been accepted as the marketed version and placed on stability? Is there a risk in cutting off analysis or trending of data?
So in my experience, especially when you're early on in development and you've got multiple different formulations sitting in your chamber, typically your risk averse choice is to trend all of them. However, I've taken the path of, once you have your final commercial formulation or something close to it, that's the one that you really try to do all of those different studies on and those different statistical analyses. That being said, I think there is also benefit if you have formulations that are close to some of your commercial formulations, or have similar properties, because that can, depending on how close those formulations are, it can also provide justification for future, I don't want to say formulation changes, but tweaks and variability within the recipe, for lack of a better word, of your product. So when you do the validation, of course, you're going to validate, a range of parameters within the manufacturing process. So again, depending on what the differences in the formulations are, sometimes you can scoot some of those obsolete ones off of your statistical to-do list. But I think it's more of a question of how are they different and what part of the process did they deviate from? Does that make sense?
Yes,
Okay.
I think so. So it's definite reasons why you can keep those going,
Mhm.
as long as they're supportive. And
Yep.
if you kill it, you probably should document well why you killed it.
Yeah. Oh, absolutely document. I mean, are you talking about the stability or the statistical analysis part?
Mostly about stability.
Yeah, if you're going to take it off, there definitely needs to be a justification, and that usually comes in the form of, maybe either an amendment to the stability protocol to say why it's being terminated at a certain time point, or or including a little bit of verbiage in the summary report that would include that.
Okay, thank you. And we'll go to Geoff Carr for another. The HPLC results reveal an aberrant peak near the main component peak that is well defined, but is only just over the detection limit. Our procedures require a full investigation, but are we required to initiate a field alert for something so uncertain at this stage, even if they won't be able to perform confirmatory testing for a few more days? And I think that's a real hardship on a field alert, because you have 3 days to get that in. So, Geoff, what do you say about that?
Okay. I think, John, you might want to do a little bit of revision on some of the procedures that you're operating. The peak just above the detection limit means it's really, really low. The detection limit is defined as a peak which is about with a signal noise ratio of about 3 to 1. This is an extremely low peak and is generally considered to be unreportable. It's not quantifiable. You need to be working with peaks at a quantitation limit. The quantitation limit is generally defined as a peak with a signal noise ratio of about 10 to 1. So you need peaks with a signal noise ratio of at least 10 to 1 to have reportable data. And that's the generally accepted way of doing it. Now, for a reportable peak or for a QL of 10, for acceptable QL, you would probably want to be able to reliably report data at around, say, half the specification limit, so you can monitor increasing levels. And that's the way we generally work. So if you've got a peak just above the detection limit, it's probably not reportable, not as a quantitative result. You could report it as below QL, quantitation limit, but you would not report it as a figure, and so it would not require any sort of investigation. Next thing is that you don't need to investigate peaks which are below the specification limit unless they're out of trend. So if you have trending data which is saying that this peak is out of trend, then you need to do investigation. But generally speaking, for impurities, you don't need to do investigations at below specification limits. So your question about whether field alerts are required, well, you don't need to do a field alert unless you have an out of specification result in the first place. So having a result which is showing the presence of an impurity, but it's well within your specification, you do not need to do any field alerts there. You only need to do a field alert if you actually have a result which is shown to be out of specification.
Okay, well, thank you very
Does
much.
that answer the question?
I think it does. We will move on to another float here. And this one's from Precision Stability Storage, an Astoriom company. We have Monica Edwards with us, and I think Ken was in the background there a little while ago too. So can you tell us a little bit about what's the greatest and latest in stability storage? What kind of challenges are you working on?
I think what we're seeing here, and we've actually seen it for about the past year, is a lot of virtual companies. So you're having a project submitted through a virtual company where their CDMO may be in Spain, and they're having it shipped over here, and then we're shipping it to a laboratory, that's a third party, and it's all virtual. So, there's no more like in-house sending back to clients, for them to do their own testing, that they are farming everything out. And we are seeing a lot of disaster recovery issues come up, about clients wanting us to reserve space. So I think that the risk-based thinking is really coming forefront with stability.
Okay. I wanted to ask a couple other questions here. How often should stability chamber temperatures and RH outputs data be reviewed? I was recalling some of my more active days, I was looking at the reports at the end of every week. Is that still something people do or should be doing?
We actually review our data every month. So we will generate a report for all of our stability chambers, and we'll look and make sure that that we recorded the amount of data that we were supposed to have for the month. Look and make sure that we did not have any deviations in temperature and or humidity, and if we did, make sure that we did notify our clients.
Very good. And what kind of backup systems are employed by stability storage CDMOs, whether it's you or others you're aware of?
Well, I know that we have, of course, backup power. We have 2 natural gas generators, but we also have a stock of spare parts in case we have a chamber go down. We can fix it quickly. And we also have a backup conditioning unit. We purposely built this facility to operate on one size conditioning unit. So we done a engineering study early on showing that, if we switched that out, there was no difference in the operation. That's kind of our last resort. Our systems are so simple that we can normally fix everything with the spare parts that we have on hand.
As a little interesting aside, the tower in your float there is I believe your liquid nitrogen. Do you have liquid nitrogen as a, I don't know, backup system somehow?
We actually have CO2 backup for our -80 freezers. So we have, we noticed during COVID that dry ice was something that was not around our area due to truck drivers, being out. And so we invested in a dry ice maker, which serves us twofold. We can ship with it, of course, but if we have a -80 freezer going down, we can make our own dry ice in the house, move those samples, until the other chamber is ready to go.
Interesting. Thank you.
Okay. And then speaking back to an earlier question about fire in a chamber and some of the ways that that gets taken care of, there is one addressed in your particular direction. Has anyone had a fire sprinkler malfunction in a chamber? We are being asked to develop a disaster plan for such an event. So if the sprinkler malfunctions, it starts wetting something down. What kind of planning is there to actually have for such an unlikely event, if in fact it truly is unlikely? In North Carolina, are you required to have sprinklers in your chambers?
No, we are not. I did a little bit of research on, having sprinklers inside, and I think that they suggest, to kind of stay away from water. See if you can use a dry system instead of, water. If that's not, something that can be easily upgraded, since it is so low of a risk, really just keeping, product off the floor and maybe, trying to store it in a way that, water can be diverted from around it until, it can be moved.
Okay. And we have a question in the chat. Are you worried about CO2 egress to samples?
No, because we have our CO2 is outside that we make dry ice with. The chamber that we do have the CO2 hooked up to just injects it when it's needed to keep the chamber from going out of spec, and everything in there is a sealed product.
Very good. I had a little bit of experience with that sprinklers leaking or else overhead pipes leaking, in that we had things that were sitting in our bins in our chambers back many moons ago, and they were floating in the water that came down. So we tried to learn a lesson from that and not have overhead pipes going by. But until we could resolve that, we put awnings over under all the shelves, which would be over all the samples to take any water away and into a trough that would take it to the floor drain to do that. But yeah, it's tricky about the requirements, because some states require you put the sprinklers in and other ones do not, and it's up to your company as to whether or not they want to take that risk. Another company in South Carolina was held up by the county inspector because they were using plastic bins, which has been the standard that I've always been used to. And they were forced to go over to wire mesh because that was deemed a fire hazard in the chamber. And we're thinking, we're not sure how your bins would set fire in a chamber, but they had to go to the expense of changing everything over to wire mesh things. So that was pretty interesting to do. Let's see how we're doing time-wise here. We have a little bit for some more questions. Let's go back to Emily. This one's about a micro lab, and I think you're probably as well versed in micro as anybody here, unless we have a microbiologist in the audience. The micro lab is telling us on day 12 that there's certain the TAMC, total aerobic microbial count, will certainly fail before the 30-day incubation period is up. At what point should we initiate the investigation, now or after the actual test completion and verification? If not now, is there some action we should take to avoid criticism? You have your microphone hat on today?
I do. So I guess my first question is this supposed to be an aseptic product?
That's a good question. We have a submitter of that question say whether or not it's aseptic. Let's say it is, just for argument's sake.
Okay, if this is supposed to be sterile, I would have some questions as to the cleaning of the equipment prior to manufacturing the batch, along with, you know, the container closure, were the components sterilized prior to that. But considering we don't have that information, regarding when to start an investigation, I think in good faith, it would be good to notify QA that you're starting to see some issues and that this may possibly fail. The actual reporting of the failure would happen after it's actually been reported, but there's no harm in alerting your management, your QA lab representative, that you're seeing some issues. This is a possibility that you may have a failure on a certain day, just to let them know that there could be a possible investigation coming. Given the fact that the way my limited micro experience, it's one of those things that it's either sterile or it's not. I would want to have either pull some additional samples to see if this is an isolated incident or if there's something wrong with this particular batch.
Okay, thank you.
You're welcome.
Let's go over to Walter. Question for you. Just going to say locate where it is. When purging samples from completed studies, this is a good one from my career, what type of accountability is expected to prove they were sent out and destroyed? Has anyone had an auditor ask for proof of destruction? And I guess a follow-up to that would be, how long is it good to hold on to samples at the end? In my early career, they wanted us to hold them forever because we had space, and they might actually need them for some reason. And we eventually ran out of space and put an end to that practice. But what are your recommendations for this one, Walter? You're muted still.
Sorry about that. Yeah, the only situation I had with this was from an internal audit where they were questioning our chain of custody, although they wanted us to prove that we could establish that chain of custody all the way to the landfill, essentially, or to the shredder, et cetera, even for the packaging. In fact, they would have us, destroy or make some sort of destruction mark on the packaging even before it got shredded and such. But I, to be honest, I've not had anything close to that kind of question from a FDA auditor or any regulatory auditor. I think, as long as things look clean and they're not seeing a bunch of garbage everywhere, they're not going to question your destruction process, especially if your inventory is accurate. So yeah, I think you should definitely have something in your procedures to that effect, and how you're following that chain of custody all the way out the door. But I don't believe an auditor is going to ask you more than beyond the door. They're not going to ask you to prove that the garbage truck really did go to the dump. So.
Just a thought I had from a digitalization position. We've had clients ask us to add in features where, because we have a complete inventory control built in, and we've added in features that they have a witness sign off at the discard point. So you're never allowed to discard samples by yourself. You always have to be two people, and both of you have to sign your electronic signature in the system to say that these samples were properly disposed of.
Wow.
And yeah, and there has been other instances of different changes we've made specifically for clients that handle, narcotics, controlled substances, and so on, where the chain of custody always has to have two people. So it's pretty interesting, and I think maybe it's really product dependent. Depends on what your product is. I mean, if it's just aspirin or something, that's one thing. But if you're dealing with, narcotics, those oxycodone painkillers, things like that, then the disposal of these is tracked, with multiple people. So just thought I'd add that.
Assumes that your municipality hires somebody to stand there at the dump and sign off on those things, which is a good reason why we could keep people employed. Let me go next to the Kennex float. Kennex doesn't have a representative here today other than me talking about them. We wanted to highlight them because they are a co-presenter of the very next program we have coming up in March about updating stability equipment and systems. And Kennex is a seminar company that really goes out of their way to make sure that stability is addressed in some of their meetings. Unlike PSDG, which is a discussion group, Kennex is more presentations and does a great job of that. They have a program coming up in June 11th and 12th in Philadelphia. And I would suggest if you can't attend a PSDG meeting, which we'll talk about in just a moment, that's a good alternative. Whereas you can pick up the how-tos with them, you can pick up the how is it going with us kind of thing. So wanted to put in a plug for Kennex and make sure that people were aware of their programs and services. And then that does take us to the PSDG, Pharmaceutical Stability Discussion Group. This flow kind of depicts the very first meeting. That's about how many people we've had. And now we've grown to over 1,000 participants over the decades. So you can see we're on our third or fourth decade here. And looking forward to a meeting in May on 17th through 19th in Wilson, North Carolina, which is a beautiful spot. And we're hoping that a number of people here who enjoy this kind of format where you can raise a question and then follow up on it and do polls and take it 6 ways to Christmas as to how it gets accomplished, what happened when an inspector walked into your stability area, what kind of things did they say, whether or not you implemented a recommendation you heard and how that went over with the inspector. And you've heard some stories here where inspectors were very critical and other times they're looking for evidence. So you want to make sure that your company, if you're implementing something, you can tell what's happened to others when they've implemented the same, whether or not it was successful. You can save yourself a lot of grief and trouble that way. And during the course of the 3 days, you'll have a chance to meet with vendors and see their wares and ask them questions, as well as interact in social situations and hear some good stories. You can ask a lot of polling questions during those days as well. And we really appreciate the fact that it's Precision, the last float that we were just at, is the one who stepped up and volunteered to be the host for this particular meeting. So they're based in Wilson, North Carolina, and we're looking forward to visiting with them. We're looking forward with seeing many of you as well. We have a limit of 50 seats, and we're 10% of the way already filled those up. So you can check out the StabilityHub website to see about registering for PSDG meeting in May. It's kind of interesting. I like watching the interaction that happens during the day. You have some brief responses from some people. And then if you revisit that same question about 8 o'clock at night where we're on our activity and 2 glasses into the Zinfandel, you get a lot more information on the same topic. So just an endorsement of that particular concept. And then StabilityHub, the organization that's sponsoring what you're doing now. StabilityHub.com is our website. And if you're new to that, we encourage you to go to the site and take a look at it. These are some of the features, newsletter. You can sign up for with the drop-down menu when you visit the site. You can check out regulations from around the world for stability, publications that have been written about stability, and articles that the people who participate here have written. And then there's a discussion forum for you to ask questions and comment on the experiences that you've had. And then a listing of events, and some of those events we've highlighted this afternoon. And we are powered by Cavitec, and I wanted to put in a plug. That's our web designer who's done a fantastic job for us. So if you're looking to establish a website, Cavitec is a great one to do. I'll take this opportunity to entertain a few more questions while we have time. And we'll go over to Geoff. And it has to do with the number of things going on stability. The required number of sample packs, Geoff Carr this is, placed into stability chambers can be easily calculated for the number of designated pull points, but did the panel recommend placing additional samples into chambers? Might this create a risk that during a regulatory audit, this might be considered as evidence of fraudulent practices? And yes, great question, because we've been accused of having too many samples, and then if you don't have enough, you can get accused of not having enough samples.
Yeah, absolutely, John. And yes, it's vitally important that when you start your stability study, you do have sufficient numbers of samples in the chambers to enable you to do enough testing as required in the protocol, and that you are able to take you all the way through to the end of the study, which is going to last probably for several years. So if this is not being done, you can be very embarrassed if you were unable to complete your study because of insufficient samples. Having said that, you can certainly easily calculate how many you need from the testing that's required. So should you put in additional ones? Well, over the years, accidents can happen. You could drop a sample, have spillages, some things like that, and you need to be able to deal with them. Also, it could be that when you take a sample out of the chamber, you might find the packaging is damaged, and we would certainly not be testing samples which are in damaged packaging because they would not be representative, so these would not be used. So certainly we always put spares into chambers so that we have enough to be able to complete the testing. So yes, now that you've put spares in the chambers, some of your friendly investigators might then decide that, well, how do we know you're not testing a sample, deciding that you don't like the result, and so you pull another bottle out of the chamber instead, and you retest that, and then you report that value, which would be a fraudulent event.
Mm-hmm.
So yes, we would put spares in the chambers, but the requirement is that if you do, for any reason, to take an additional sample out of the chamber for whatever reason, you're going to open a deviation record. You're going to give the full details of the dates and times, what sample was taken, how many bottles you should have left will be entered in your requisition documentation. And the reason for it will be given in the investigation report. If it requires corrective actions because somebody was clumsy, then that would also be done. But the important thing is that you have a deviation record which is available for an investigator to inspect. And so full reasons for use of those samples are provided. In that way, you are clear that you're not doing fraudulent activities, and that's the way to proceed, and you will always have enough samples to complete your study.
Thank you, Geoff. Hey, we've hit almost the end of our time here. And just like Santa Claus appears at the end of the Macy's Day Parade, Svante Arrhenius appears at the end of the Stability Day Parade. And he actually waves to the crowd. And wanted to take this opportunity to thank our folks that have been with us for the panelists. Get on to there. That includes Geoff Carr, Pharmaceutical Consultant, Geoff Cook, Parameter Generation and Control, a Binder Company, Susan Cleary, Novatek International. We didn't see Adam Margolis, but we thank LLab for sending some materials. Emily Dickinson, Linical, Monica Edwards, Precision Stability Storage, Maria Krisch, FreeThink Technologies, Chris Latoz, and Walter and I. We thank you